I'm going to be going over reprocessing validations, and I know a handful of you guys do work with that. Oops. Okay, so these are some of the devices that we counter with reusable medical devices. When I talk about reusable medical devices, a lot of people think that these are surgical trays or endoscopes or things that are invasive. But usually they are things that are in the OR room. Surgical lights, believe it or not, we've validated a surgical light as half of this table is, right? Because if you're in procedure, if you cut open somebody and the blood splatters up there, how can you assure that that's clean and and it will not cause cross-contamination, right? Hospitals have internal procedures of how to clean all these type of devices in the OR room. However, are they validated? They're probably not. And so they go to the manufacturers and ask them. This is capital equipment that is placed around in the OR, or it's used for devices that are invasive. So we have seen an increase in the last 2 years on these type of devices that have come through for validations. Now, Lisa went through thorough cleaning. A lot of people see when we talk about reusable and reprocessing validations, it's one step in the healthcare facility, right? Point of use, like Henry said, cleaning, either it's disinfection or either it's sterilization. So it's one process in the hospital. Then why aren't we validating a one process for the validation? Why do we break them up in pieces? The reason for that is we're looking at end markers. Like Lisa said, we're looking for removal of contaminants that are on the device. That's protein, hemoglobin, TOC. These are endpoints that we're using as markers to test. For disinfection and sterilization, we're looking at microbes. So we're looking at a kill. And we're looking for a kill log reduction for each one. However, they're different. When we look at thorough cleaning, we either choose the next step as sterilization or disinfection. It doesn't go all together, except in Europe. Here we do all of them, but in the United States, it's either one or the other. We don't do all of them. I'm going to go through the disinfection validations and how we perform those validations in a little bit more detail so you understand what's entailed in these validations. Now, why would you use disinfection and not sterilization? Disinfection is more applicable to devices that cannot withstand high temperatures. And also devices that are sold in countries that don't have sterilizers in place or clinical areas. A lot of third world countries, they can do the manual cleaning, but if they don't have a sterilizer, an autoclave, how are they going to decontaminate? And even if it's a critical device, many countries will put it through a high-level disinfection because at least it's some sort of a safety margin that they're adding to that. So that is when you would choose one or the other. I've got a question mark in Europe for the reason that thermal disinfection is something that is almost required in Europe for healthcare users. It's not for your device, however, it's for the users, the end process. So when it goes to the automated washer, like Lisa talked about, the other side, the users are pulling them out and getting them ready for sterilization, right? So when they're doing that, they want to make sure it's safe for the users to handle the product and get it in packaging for sterilization. Thermal disinfection is also done if that is your end point for validation. And there's no sterilization needed at the end. There are 3 methods. There's manual disinfection, automated, and thermal disinfection. For manual, we're just basically soaking the device in the chemical for the specified time and temperature specified by the labeling. For automated, this is more we use for endoscopes, automated endoscope reprocessors, or other machines that we have that will chemically disinfect the device. And then there's thermal that we use through automated washers, either through ampules or through microbial testing. And there is a difference between those validations. If you're going to submit thermal disinfection as your end process, you have to do two validations. One is through either an ampule or a direct inoculation method for US FDA, because they do not recognize the thermocouple testing, the temperature profiling testing that we do. And for Europe, you will have to do temperature profiling, as they will not accept any microbial testing for it. So unfortunately, in that terms, you will have to do two validations at that point. Now, when deciding what sterilization method is appropriate, it depends on the Spalding classification. There are three sets where you would have to figure out. There is critical devices. These are invasive devices that are going through the body and going through the tissue. Those have to be sterilized. Now, if you can't sterilize them, you can do HLD. However, you better have a good justification on why we're doing high-level disinfection on a critical item that should be sterilized. If your device fits in semi-critical, which is it's contacting mucosal membranes, then you want to do high-level disinfection. And then for non-critical devices, these are devices that touch the intact skin or is cross-contaminated during use. So if I'm a nurse and I'm helping a surgeon, but say if I touch something like this, now I've cross-contaminated that. That will have to be validated. So that's when we will decide if it's an intermediate level disinfection or a low-level disinfection method. They each have their designated bacteria, microbes that you have to use for validation, and designated log reduction values that are required to show that you have shown the efficacy for the category that you're claiming your product to be. These are some of the examples that I have put up here that so you can see what an intact skin device would look like for mucosal membranes and sterile areas. Now for ASIB, for washer disinfectors where we're doing temperature profiling, we do have their own specifications as well. So for invasive devices, you want to see greater than 3,000 ASIB, or microbial, you would have to do a high-level disinfection where you would inoculate with Mycobacterium terrae to look for a 6-log reduction, and so forth as you go down. Now, we can't do this validation without controls in place. So you have, like Lisa said, we have positive controls. We don't need a negative control in here because we're looking for an indicator organism that we have contaminated the sample with. You only need 3 test articles. That's all you need to show disinfection on. As long as you show 3 test articles, this disinfection validation is good for life. If you change your product, if you change your materials, then you have to revalidate again. But until then, you're fine. You have to do a neutralization process, because if you don't neutralize, I don't know if my extraction ileum that I'm using to extract the devices will cause a problem with the bacteria during incubation. So we have to do a neutralization process and cytotoxicity. We do cytotoxicity testing to ensure that the chemical that we've used to disinfect the product will not adversely affect the patient. And there has been debate, 1 or 3, what is the right sample? If you do 1, some regulatory agencies are accepting it. If you do 3, it's also accepted. So, so we're in a limbo right now to figure out what do we recommend. Do we recommend 1? Do we recommend 3? Where are we setting. So we are obviously going to be in the default and recommend that let's do 3 so you don't get a kickback from the regulatory agency. Like cleaning validations, we have to contaminate the devices with bacteria, and we do test in a mixed suspension, except for high-level disinfection, and we inoculate in areas that are most difficult to sterilize. So we're just placing droplets of inoculum in areas that we think will be most difficult to sterilize. So it is direct inoculation method. The different phases of disinfection validation include, like I said, neutralization, cytotoxicity, and bioburn testing. And this is an example of mixed suspension plating that we've done with selective differential agar. This is McConkey agar that we use to evaluate Klebsiella pneumoniae and E. coli. And you can see they're clearly distinct, so we can test them together as one piece. And these are validated to ensure that they can grow up easily. The one thing to remember about disinfection validations is if you're submitting anything to US FDA, you have to choose a high-level disinfectant from their 510K approved high-level disinfectants through their website. I've got the website right here that can go, if you go there, it will pull up a whole list of disinfectants that are approved by the FDA to use as a chemical sterilant or a high-level disinfection. Oh, let me just go over. You will see that there is not a huge selection. However, glutaraldehyde solutions is a dominant disinfectant that we use in US. However, in Europe, a lot of the countries have banned the use of glutaraldehyde solutions or do not favor towards them. So we do see hydrogen peroxide and peracetic acid, or hydrogen peroxide and peracetic acid together, validated for EU. So we do have 2 different disinfectants sometimes validated. Now, when you're doing intermediate level disinfection or low level disinfection validations, it is recommended that you pick disinfectants from the EPA approved. We have seen US regulatory agency asking for an EPA number of the disinfectant you used. Not all do that, but some do that. So we always keep stuff in-house that's EPA approved, so we don't have to go back and revalidate that if we don't have that information. A lot of these disinfectants are either spray or wipe. So the difference between high level and intermediate and low level disinfections is high level is you're immersing the product in the chemical, while for the others, you are either spraying or wiping. And for low level disinfection, which just contacts the skin, it's just a wipe, and you're trying to show 6-log reduction with that. The most difficult disinfection validation are these. Can anybody tell me why? So I'm touching it, patient contact. I put 1 million bacteria in here. Now I have to wipe. How is the hospital going to wipe? Done, next patient. I can't do that. It's not going to work. We're not going to get a 6-log reduction by just doing that. So we're going to be doing a validation where it's going to take much more than that. I'll have to either wipe it a couple of times, have a wiping action, removal, and that's how we're going to validate it. Hospitals are definitely not going to follow that, and I think today we've learned that whatever we validate is just for us and just for books, and for the regulatory agencies, what happens in the healthcare is what happens in the healthcare. We just have to look the other way sometimes. And who was it, you, Henry, who said that when you go to the healthcare facilities, the IFU is just tossed? I have seen that myself. And when I've gone to the CSD departments to to just audit, and we'll ask them, where are your IFU? They'll be like, oh, it's in the basement. Where the supervisor is, it's in the cabinet. So I'm like, how do you know that you're following these procedures? And how do you know what you're doing? They're like, oh, here's a binder. There's 8 pages in the binder. And they have categorized the device. With lumen devices, you do this. With drills, you do this. With hinges, you do this. And they just have those 8 procedures that they're following. But that's the reality of healthcare facilities. Like we discussed before, US, we're looking at glutaraldehyde solutions, and EU, we're looking at the hydrogen peroxide or parasitic acid solution. So unfortunately, if you want to do market in US, hospitals might say we're not going to get the parasitic because that is more harsher on the product, and we'll use something glutaraldehyde, and they'll probably ask you for that validation. So most often we see manufacturers do both of them. Now I'm going to talk about steam sterilization, and why steam sterilization is because it's the most predominant method of sterilizing reusable medical devices. Why? It's fast, it's reliable, it's dependable, and it's non-toxic. And in a healthcare environment, you have to turn around these devices very, very quickly. You can't just clean, sterilize, and shelf it. It hardly goes on the shelf. It's going back to the OR for use. So this is the fastest way to do it. However, there are other methods. We do have EO for reusable. For endoscopes, we can only do EO. So we have to do EO validations for that. We do have VHP. However, not all hospitals have VHP. But all hospitals do have a steam sterilizer. So that's why steam sterilization predominantly takes most of the reusable medical device industry for sterilization. Now, believe it or not, I've got all these stuff mentioned here, and I'm sure when you guys are doing your risk assessments, you know what to look for. But every year we get one person that sends us the device, we look at it and we advise and say, I don't think this is going to go through the sterilizer, it's probably going to melt. They're like, no, we've done our risk assessment, I think it will work, let's just put it through one cycle and see. And so we trust, we're like, okay, they've done their risk assessment. We put it through, believe it or not, comes back stuck on the sterilizer pouch, and we can't open the pouch, and it's melted. So always make sure that if you are designing a device, it can withstand the temperatures. We're looking at 121 to 135 are the temperatures we're looking at that it should withstand. How you design a tray for steam sterilization, because a lot of them are surgical trays, is very, very important when you're validating these. Materials you use, the brackets you use, how you're placing the brackets within the tray, where you're placing your devices that have lumens. Are you placing them on the outside? Are you placing them in the middle? Because in the middle it's not going to work. Is it on the bottom of the tray? Is it on the top of the tray? Where do they belong, and how do they belong? Very, very important in steam sterilization. Also, using less than 25 pounds is in ST79, which is a standard, and in US, it's a requirement. If your surgical tray is more than 25 pounds, they will ask you to split them in two. So you will have to maintain that 25-pound criteria for your surgical trays. Now, when it comes to sterilization, like Lisa said, you probably could do for cleaning, you can do family grouping out of the sterilizer, or pick a worst case instrument out of the sterilizer and do the validation. However, for the steam sterilization, we will use the entire tray and validate. We just need one tray that we'll do 3 cycles with. And for devices, we would need at least 3 devices with the 3 cycles. For the steam sterilization validation, the most resistant organism is Geobacillus stereothermophilus. We do use the overkill method approach. And what that means is we're inoculating the device with 10 to the 6. We don't do the bioburn approach for reusable medical devices because bioburn will be different per person, and we can't estimate that. So we do take precaution and do the overkill method for that. How this is determined is we place BIs in the most difficult locations to sterilize, lumens, mated surfaces, cannulas, any heat sinks. If we have a tray that is very, very filled, where you have instruments, we would put it underneath that area if there's a caddy in there. So that's how we would do it. There are 3 steps to steam sterilization validation. The first step is we're going to do a sterilization level validation with half-cycle parameters. So we're going to inoculate the devices with 10 to the 6 and ensure that in the half cycle of that entire full cycle, it does kill a 6 log. The next one is the dry time validation, where we do a full cycle validation for that, and then temperature profiling. For the SAL validation, we're looking at we're selecting locations in the tray or on the device. That's called product inoculation. We package it, program the half cycle on the autoclave, do a sterility test, and then score for growth. Very simple steps. For product inoculation, we're using BI strips. We're using that you can see down here that we've placed underneath a caddy. We are inoculating with sutures and wires. And if you see that we pulled up a little screw from here, put it in that, and then we'll put it back in there. So we are trying to find locations that would be most difficult to sterilize. And usually this is something that we work with the manufacturers to determine, here's our tray, and we will help pick out the worst case sites and talk to the manufacturer to see, do you guys agree with us? Do you have other sites that you want to test? And so this is all discussed pre-testing with the manufacturers to make sure that they feel that's appropriate. Once you have inoculated, you're going to be using packaging. There are 3 types of packaging for sterilization. We use the cellulose pouch for lightweight devices. You don't want heavy devices because this is a steam sterilization. It can be wet, and if it's heavy, it can damage and breach the sterilization barrier. Sterilization wrap is what we use for sterilization kits or container systems or even devices. And rigid containers, not often used, but they are out there for validation as well. So once it's gone through the sterilization process, we will remove the BI strip and place it in a growth medium for growth, and it's basically a visual assessment. It's either it grew or it didn't, and that's how we score for growth. This is what you would see in your final report, is that it was there or it wasn't there. For dry time validation, you have to prepare the packaging that you're going to use. So if you're using a big surgical tray, you want to cut it out. You want to make sure that it's the right parameters for the tray. You would weigh that. You would package the devices, run a full cycle on it, remove the packaging, and reweigh the packaging again. And here is the specifications for it. Packaging weight cannot exceed 3%, or if there's any visual soil. Believe it or not, anything that's heavy and what we call heat sinks are devices, tray surgical kits that have rubber mats. Those are problematic. That's where you'll see moisture build in. and will fail the product. So there's a lot of things that you have to consider when you're putting these trays together, especially even if you're using any plastic material. If you see condensation, that will fail the product as well. So we're looking at not only the packaging, but the internal product as well to ensure that there is no water droplets anywhere. The last phase in sterilization is temperature profiling. This is where we would program the temperature probes, place it, and run a full cycle, remove it, and run the data. And when we run the data, this is what we're looking at to see how well the temperature profile was in those locations. And we usually choose locations that are cold spots in the tray or within the device. The two methods of sterilization is gravity. Now, gravity is something that was something that was used a lot. It's being phased out. Why? Longer cycles. Nobody has time to do 15-minute cycles anymore. So it's being phased out by healthcare facilities. But pre-vacuum cycles, which are shorter cycles, where we have pulses of steam that goes in, suction out, 3 to 4 pulses that we're looking at that are going in and out. And that's what's most commonly used in healthcare facilities and here and in US. So pre-vacuum sterilization is your most common sterilization method that you should think about and use. However, dental places still use gravity cycles. So if you're designing devices for dental, you may want to talk to that audience and see if they do have pre-vacuum cycles or they're still doing gravity at that point. The one thing that is very important with these modalities is that you want to use FDA-approved sterilizers. They will ask for that information. What is the 510K number of the sterilizer? FDA-approved accessories and biological indicators. And parameters are also very strict. If you don't fit in the parameters, like we have here, 132 for 4 minutes for US, it won't be accepted at all. You will have to fit in that, no matter what. That's why extended cycles are not looked at. They will not accept any extended cycles unless you have a justification to why your product needs an extended cycle. They do look at justifications, but they do discourage for extended cycles. This is the cycle for US that you have to meet. And this is something that when I went to a presentation, I put data together that in EU, this is what's commonly used in different countries for steam sterilization cycles. So there is a variety. There is a difference. We only have one cycle that you have to meet. But here you have different cycles that you have to look. So you have to do your risk assessment to figure out what's the best modality, what's the most used cycle time. If you are in the reusable world and you haven't come across this standard, very, very important standard for US. All the validation methods for cleaning, disinfection, and sterilization and repeated use are in these standards. Are in this document, and EU also will follow this as well. So something to be familiar with. The rest of the slides are just references for reusable medical devices. You'll be amazed how many we have. We have tons of them, but look at that. I got you at 5 o'clock. Thank you.